The broadcast is now starting. All attendees are in listen-only mode.
Hello, everyone, and welcome to today's webinar. This is Christy Sullivan, and I'm the Secretary of the American Society for Cellular and Computational Toxicology. This webinar, like all of the webinars in our series, is sponsored by ASCCT as well as the European Society for Toxicology in Vitro. We collaborate on these webinars. Both of our organizations hold regular meetings and pursue other activities to promote computational and in vitro toxicology. And we, in particular, offer training and education and try to support early career scientists. If you want to learn more about either of our societies or want to become members, we would certainly appreciate the support. You can visit our websites here on the screen. I also wanted to give a little bit of housekeeping. The webinar is recorded, as you heard, and the recording will be posted on the ASCCT website in a couple of days. And you'll receive an email afterwards with a link to a PDF copy of the slides. Also, all of the lines are muted. There will be an opportunity to ask questions after the presentation. In fact, during the presentation, if you want to enter your questions into the Q&A box, please do that, and then we'll have a Q&A session after the presentation. And then if you have any technical issues, you can enter communication in the chat to reach me, and hopefully you'll enjoy the webinar. So I just wanted to briefly mention that the ASCCT just had its 9th annual meeting in October, and we had a virtual meeting. There were over 200 around 200 participants, 70 posters, 20 oral presentations. As you can see from this poll, which we conducted during the meeting, people from all over joined us, so the virtual format really helped us to gather more audience members. The recordings and the posters of during the meeting are available to members and to registered delegates on our website. But you can also, if you're not one of those folks, you can also go to the annual meeting page and find out a little bit more about the meeting. And you could volunteer as well to help us start to plan for our 10th annual meeting in the fall of 2021, and hopefully that will be in person. I also wanted to mention that there are a couple of upcoming webinars. We've managed to squeeze in a really exciting update on alternative approaches for medical devices on December 17th. Before the end of the year, and then January 14th, we'll have presentations from 2 of our award winners from the meeting, some of our outstanding student presenters giving talks about their work. Neither of these are quite yet available for registration. We're just pulling the registration pages together, but those should be available really soon. And so you can visit our website to register in the next couple of days. Or the best way, besides becoming a member of the society, is to sign up on our website for email notifications, and you'll get occasional notifications, including upcoming webinars and things. I also wanted to mention, just to put in your mind, for 2022 is the next ESTIF Congress. They had been planning their Congress in June of this year. However, it had to be postponed due to the COVID pandemic situation. So you can visit their website to find out more about that meeting. And with that, I will move on to today's presentation. So we're going to hear from Dr. Adam Lillicrap from the Norwegian Institute for Water Research. Adam is the research manager for the section of ecotoxicology and risk assessment, which employs 20 dedicated research scientists and several PhD students. He's also NEVA's GLP manager, has been project managing and study directing regulatory ecotox tests according to GLP for over 20 years, and is a Eurotox registered toxicologist. Adam is the Norwegian representative for the OECD Validation Management Group for ecotoxicity test methods and for ISO water quality test standards. He's been working with zebrafish embryos for ecotox assessments for almost 20 years and was nominated expert to the OECD regarding the implementation of the fish embryo tox test guideline, and then more recently, the RT rainbow trout gill assay at the OECD as well. Over the years, he's been very active in the field of animal alternatives for ecotox, chairing sessions at CTAC, and promoting the implementation of these tests at the regulatory level. And finally, Adam's currently leading a CEFIC LRI project entitled Strengthening Weight of Evidence for FET Data, in which he will be talking about to us today. And I think rather nice for a researcher, Adam has a thorough understanding of regulatory complexities regarding adoption of alternative test methods and the need for a weight of evidence approach. I'm going to go ahead and give you the floor now, Adam. And
thank you, Christy.
Let you share your screen, make you presenter. Let's see. Okay. Should be able to share your screen. Yep, I can see your screen full screen. So please go ahead. Thank you.
Excellent. Thank you, Christy, and thank you for the introduction. Sounds like something I would have written myself, actually, but very good. Yes, so thank you. I'm Adam. I'm the research manager for NEVA, the Norwegian Institute for Water Research, and I'm here to talk about animal alternatives. So I've brought my Save the Fathead t-shirt, which I wear with pride most often, but not today. I'm wearing my NEVA fleece today because it's a bit cold outside. Okay, so I'm here to talk about the fish embryo test. I really perhaps don't need to indicate to people about the 3 R's, because I assume everybody here should know about what the 3 R's, the reduction, refinement, replacement, and hopefully most people will know that it was first brought as a concept by Russell and Birch back in 1959 in their Principles of Humane Experimental Techniques book. So this is a picture of Russell and Birch. I normally, when I talk about alternatives, include this slide, but mainly because it's not just these two guys that have been working with animal alternatives. And forgive me here, I'm talking about very much an environmental focus rather than a human safety focus. So these people are all the people that I've been working with over the years, who all have worked extremely hard trying to get animal alternatives approaches accepted within for environmental ecotoxicity testing. And maybe you are aware of these additional 3 R's, and this is again one of the things that I always try and bang on about, is that it's not just the traditional 3 R's which are important, it's these additional 3 R's which actually are becoming much more required in terms of how we get animal alternatives accepted. And as you can see, there's one which is clearly shown bold, is the idea of getting regulatory acceptance. And we can produce all the alternatives we want in the world, but unless we get regulatory acceptance, then there is really no point in these assays that we develop. And there's a very nice focus piece that was in the Environmental Toxicology and Chemistry journal discussing about this idea of the 6 R's, as we also call them now. Okay, so I'm here really to talk about the fish acute toxicity test and possible replacements of that with regards to other alternative methods that we may have. Now, the fish acute toxicity test has been around for a very long time. It's one of the base set endpoints that's required for environmental toxicity testing. And there are several different species that are available to be tested, one including the fathead minnow, obviously. But most often the tests are performed with the likes of rainbow trout. So this is what a typical fish acute toxicity test looks like. It's not that exciting. It's just a series of tanks with fish in them. And the test is 96 hours in duration. And basically the endpoint is here to look at what concentration kills 50% of the fish. So a pretty crude endpoint. So we typically have 5 concentration plus a control and a solvent control if necessary. According to the OECD 203 guideline now, you can have a minimum of 7 fish per concentration. You can also use a limit test where you can base your concentration on the most sensitive of the other base set organisms which are required for ecotoxicity testing. So that is the algae, the 72-hour algae growth inhibition test, or the 48-hour Daphnia immobilization test. So what you do is you set your concentration based on the most sensitive of those 2 endpoints, and then you only need to have 1 test concentration. You could also use the limit test at the limit of solubility if there's not going to be expecting any toxicity at that. As I said before, it's 96 hours in duration and death is the endpoint. So it's a pretty crude test. So what are our options? Well, we have this fish embryo. Now, the fish embryo, many people might know about fish embryos. This is actually a zebrafish embryo, and it can be used in toxicity assessments. And going back as far as 1994, Schultz and Nagel first proposed it as an alternative to the acute fish test. At this time, it was a 48-hour test, and it was submitted to the German Federal Agency of the Environment as a test to be able to be used for effluent testing. So already we're looking at quite an old test. And why can we use the fish embryo test? Well, according to certain legislation, and I know this isn't necessarily the case for the US, but within Europe, the embryonic stage of development is considered to be a non-protected life stage. So they're not counted as a number. So in this graphic here, anything on the left-hand side, or my left-hand side, it might be your right-hand side, the green side is the embryonic stages of development. And this is anything from the egg to being fertilized, the developing embryo, up to the point of being where it hatches, but where it is still not capable of independent feeding. So it still requires the use of its yolk sac. Now, this is a relatively gray area, and some people will argue about what constitutes being independent feeding. But for the time being, we'll just focus on it being the point after hatch. And then after that, you've got the free swimming and feeding larvae, which then become a protected life stage. So any of these parts in the red area are areas to be replaced, reduced, or refined in some way. So this is another picture of a zebrafish embryo. This is an early stage development of a zebrafish embryo. But why do we want to use them? As I said before, they're not classed as protected. They have several other benefits, such as they require small volumes. We can use them to look at genetic effects. A lot of the genome is used within embryogenesis. So if there's an embryotoxic substance, they're a very good model to be able to use. Most are completely transparent, so you can monitor the development of the embryos. Generally, they are comparable to acute fish toxicity data. We'll get back to that a little bit more a little bit later on. But some toxic metabolites may not necessarily be synthesized in the egg. There's also the possibility that some chemicals, such as really large polymers, may not necessarily cross the chorion. So in terms of barriers to entry, we have several parts to the embryo which potentially would stop these large polymers going through. So you have the chorion on the outside, the egg shell. You have the perivitellin fluid as well, which offers some level of protection. And then you have the multinucleated yolk syncytial layer as well, which also gives some level of protection. Okay, so Schultz and Nagel first proposed this test back in 1994, and then it took a while for it to be put through international regulatory guideline acceptance. So already it was being accepted within the national level in Germany. It wasn't until 2006 that there was the first version submitted to the OECD as a proposed new test guideline. At this stage, it was still a 48-hour test following the original design that Schulte and Nagel had proposed. But what I really want to highlight here is that it was clearly stated that this test was proposed as an alternative test to the fish acute toxicity test. So this test is relatively straightforward. It's not a much more difficult than running a normal fish acute test, apart from the fact it's on a much smaller scale. You also have slightly higher number of replicates, and there is a lot of statistics that went into this to design the test appropriately to be able to have enough power from the test. And in this diagram here, we have 5 concentrations, each of the plates representing a single concentration. You also have 4 internal controls within each plate. Then we also have a negative control, so the NC, a positive control being the PC, and if necessary, a solvent control as well. So the test had been submitted towards the OECD. It needed to go through a significant amount of validation before it was accepted. And to my knowledge, I think it has had the most rigorous validation of any of the test methods, particularly within the ecotoxicity world, than any other test guideline that's ever been accepted to the OECD. So there are 2 reports that were written about the ring trial that was performed, and this was on a vast number of compounds. And then there's a pretty nice paper that was written about the validation study back in 2014. So after we'd done the ring trial at the OECD, we finally got the test accepted. So this is from 2006 until 2013, and anyone who's been involved with OECD guideline development knows that it's not a quick process, and this is a perfect example of that. But what I really want to highlight here is that one of the problems was that to get this test accepted, irrespective of the ring trial, there were some member states who did not agree that it should state that this was an alternative to the fish acute toxicity test. To get this test guideline to be accepted at the OECD level, it had to be removed from the introduction. So now, unfortunately, we have no reference to this being a direct replacement to the acute fish toxicity test. So we've got our test guideline. Now we want to get it regulatory accepted. So this is where the European Chemicals Agency got involved. And they did a pretty comprehensive review of the comparison between the FET test and the acute fish toxicity test, which we often refer to as the AFT test. So this is where they did a big review of all the data, making comparisons between the fish embryo test and the acute fish toxicity test. But in life, as we all know, nothing is ever simple. There is never a perfect one-to-one correlation. And this one little substance, allyl alcohol, ended up being a problem for everybody. Allyl alcohol is not metabolized to its toxic metabolite, which is acrolein, within the zebrafish embryo test. Whereas in the juvenile acute fish test, it is. So there is this outlier, or one of the outliers, which can show that basically the FET test is not as sensitive as the acute fish toxicity test. There are other substances which go in the opposite direction, where actually the fish embryo test is more sensitive than the acute fish test. But for this reason, it was not considered to be as protective as the acute fish toxicity test. So this causes a problem. But in a little-known guideline in at REACH, at the ECHA, the European Chemicals Agency, for REACH, there is this practical guide for small to medium enterprise managers and REACH coordinators. And in this document, it clearly states that the OECD 236 fish embryo acute toxicity test is an alternative to the standard AFT test and can be used within a weight of evidence approach. together with supporting information justifying the reliability and adequacy of the test. But what is a weight of evidence? Now, I remember a long time ago, a good colleague of mine, Wayne Landis, said to me that a weight of evidence is just a poorly described Bayesian network. So we wanted to try and develop a weight of evidence through a Bayesian network approach. Now, I'm sure there are a lot of people in this audience who probably know much more about Bayesian statistics or Bayesian networks than me. So if there are any of the difficult questions, I would like to take those back to the people who are the experts behind this, and we can forge some dialogue later. But for those who are probably as naive as I am, or was at least when we started talking about Bayesian networks basically are a web of nodes linking cause and effect. They're linked using conditional probability tables, and I'll talk a little bit about some of these conditional probability tables later on. And we have these things called parent and children nodes. So in our preliminary fish embryo toxicity Bayesian network we developed, we had 4 lines of evidence. The first line is based on physical chemical properties, the second line on chemical categories, and this is where we have a list of compounds from Procter & Gamble that they supplied us with a very nice database with AFT data and fish embryo toxicity data for multiple different substances, but then we could link the different chemicals into categories so that we could use some sort of rudimental read-across approach. The third line is where we used other taxa, and I'll explain a little bit more about that. And the fourth line is obviously where we have fish embryo toxicity data. And all of these lines put together should be able to predict the acute fish toxicity. All right. So this first line of evidence, the line about the physical chemical properties, what we wanted to do here was, and with all these nodes and lines, we really wanted to make sure that anything we were using for our Bayesian network was pretty much regulatory accepted. So we based our physical chemical properties on QSAR data, quantitative structure activity relationships, and here's where we use the US EPA database, the EPISuite database for predicting the acute toxicity. And then we also wanted to look at whether or not we could think about, is there any way that we could predict that the compound of interest wouldn't necessarily cross biological membranes? And the hypothesis being that if it didn't cross the biological membrane, it wouldn't go into the cells of organisms and wouldn't necessarily cause toxicity. So here we used hydrophobicity as a surrogate for water solubility. And we used a cutoff value of 5.5, a log KOW of 5.5, based on the OECD 305 bioaccumulation test guideline, which uses that as a cutoff for when you should not be testing via an aqueous solution, and you should be testing via dietary exposure because of the bioavailability factor. Also, then we looked at molecular weight. Again, this is also considered to be a pretty much, I wouldn't say guaranteed, but a regulatory accepted cutoff value, and particularly at the European Chemicals Agency, the guidance documents there use this as a cutoff value to be able to predict whether or not there's going to be limited membrane crossing. And from those little points that we've put together in line 1, we can make some assumptions towards whether or not we can predict the toxicity to juvenile fish from the physical chemical properties. Okay, so I'm not going to show all the conditional probability tables, but this is probably one of the most easy to understand, I think. Some of the conditional probability tables, we're looking at 5x5 groupings, so there's a lot of information that's going in there. But in this one here is pretty straightforward. So on the left, we've got the hydrophobicity in terms of log KOW. And like I said, we have a cutoff value of 5.5. And then the molecular weight we're using as a cutoff of 600. So if we have a log KOW less than 5.5 and a molecular weight less than 600, we have a pretty high probability that the compound of interest should be able to cross biological membranes. Now, I know that there are a lot of people who are going to say, well, that's a bit simplistic, you know, and there's a lot of other factors involved. Yes, there are, but in this case, this is what we're using in terms of our conditional probability table. And then conversely, on the other side of things, if you've got a log KOW greater than 5.5, it's very unwater soluble. We've got a molecular weight greater than 600. We're predicting it to be a pretty large substance. The likelihood of it passing through the membrane is low. Okay, so then the second line, this is where we were looking at chemical category, and I did just briefly talk about this with the Procter & Gamble database. So we've basically categorized the different chemicals so that we can make some prediction. If we have AFT data for a chemical which is in a similar category, then we can make some predictions that we can possibly do read across from that substance. The third line is where we look at the different taxa, and as I said earlier, as part of the base set ecotoxicity database data requirements, we need to have a 72-hour algal inhibition test, and we need to have a 48-hour Daphnia acute toxicity test. So we should already have this data, irrespective of whether or not we've got the AFT or the FET data. In terms of the base set, we need to have these as a minimum. And as I said before, with the threshold approach, we should be looking at this data to see whether or not we can set a limit test that will require only one concentration for the AFT data. But for this purposes here, we are just using it to inform on whether or not we think we can predict on the AFT data from the algae or the Daphnia data. And then below that, we have this idea of being able to compare the algae and the Daphnia data. So we calculate a ratio based on the Daphnia and algal toxicity data. And if there is a difference, then we can in some way be able to assume that there may be a specific species mode of action that's occurring. So for example, if the algae is significantly more sensitive than the Daphnia, we could think it potentially having a herbicidal mode of action, which may not actually affect the fish. Conversely, with the Daphnia, then if the Daphnia is more sensitive, then it may be something which may be a specific mode of action, which could be affecting insects, etc., and may not necessarily cause toxicity to fish again. But if we have a similar sensitivity from both the Daphnia and the algae, then maybe these data then can inform more clearly, because there may not necessarily be a specific mode of action that's occurring. And then the fourth line of evidence, obviously, is the toxicity to embryo. And this is where we just have the embryo data. And then we pull this all together, and this is our initial Bayesian network that we've developed. And if I just show you this, this gives an indication for anyone who is more knowledgeable about Bayesian networks than myself, then they'll see that they've got the children and the parent nodes and how they're putting together in terms of the conditional probability tables to give us some prediction. And on our first initial Bayesian network, all lines of evidence here are weighted equally. And this is just to give you an idea, and this is only a small part of where we were looking at some of the CPTs for the different areas. So this is particularly related to the other taxa. Now this row of data goes significantly down. And I don't really want to go into too much details here, but for example, you can see where the black text is specified. This is where we have a specific mode of action indicating that the algae are more sensitive than the Daphnia, and then therefore it is unlikely that the specific mode of action will have an effect on the fish. So from our preliminary FET Bayesian network, we had an initial subset of 159 chemicals, and for those chemicals where we were looking at a broad number of substances, we could correctly predict for nearly 70% of the compounds, and then for a certain level there is a little bit either over predicted or under predicted. And then we reduced the number down to 106 chemicals. We were getting 70% accurate predictions. And then right down the bottom is when we went down to 20 chemicals. Now, the reason we went down to 20 chemicals, because the PNG database obviously contained a lot of information. But what we really needed to be able to do to be able to predict accurately or to evaluate the model accurately was to make sure that we had a minimum of 3 fish embryo toxicity data points and 3 acute fish toxicity data points. So this is where we're looking at much more data-rich compounds. Some of the other chemicals did not always have very good data coverage. So these 20 compounds were what we saw as being the most valid ones to be able to look at. And when we looked at those 20 compounds, we were getting 80% of the time correctly predicted, and only 20% was underestimated. So these are the compounds that were in the 20 final list. And you can see on the right-hand side that this is where it gives us whether or not we had a correct prediction, either being yes, or incorrect prediction being an N for no. And as you can see here in the parenthesis after the N's, there is a D or an A. So on 3 of the cases, the Daphnia is actually most sensitive, and on 1 of the cases, the algae was the most sensitive. Just take a quick drink. Okay. So we were fortunate enough to publish 2 of these papers based on this original Bayesian network. One came out last year, one came out this year. So these should all be open access. So if anyone would like to look at them in more detail, then please do so. So this leads us to where we are now. So we were fortunate again to get some funding through the CEFIC Long Range Research Initiative, which is mainly a lot of the European chemicals companies in Europe who are part of CEFIC, and they fund projects each year. And one of the projects was about how can we get this FET test actually accepted on a regulatory framework. So we were awarded this project this year, or last year, early this year, sorry. Time goes funny in these times, and it's called Strengthening Weight of Evidence for FET Data to Replace Acute Fish Toxicity, and the short acronym is SWIFT. And the idea being the objectives were to integrate the FET into a weight of evidence to inform on the AFT, to develop an approach that provides improved weight of evidence on acute fish toxicity from the FET data, develop a means to address non-conformance of FET predictions, so those compounds which may not be metabolized in ovo, identify additional gap filling needs and develop short and long-term priorities of addressing these needs, and to develop additional lines of evidence, including mode of action, phys-chem properties, metabolism, gill cytotoxicity, neurotoxicity, threshold determination, and additional sources of data. So in a simplistic way, we've got our original Bayesian network, and now we're including metabolism as a new line, QSARs where we can pull in additional QSARs, the gill cytotoxicity assay, and as Christy indicated earlier, this is now an ISO standard, so it is a regulatory accepted method as an ISO standard, and it's also being evaluated at the OECD test guideline program. And then we also have neurotoxicity, being that neurotoxicity can be a bit of a problem with fish embryos. So the gill cytotoxicity assay, this new line of evidence, this will be its own line of evidence. And there are 3 endpoints which look at cell membrane integrity, lysosome integrity, and metabolic activity. And this gives us some information about the cell viability after exposure to these compounds. And then we can make some assumptions towards the toxicity to the fish based on the cytotoxicity assay. Then we have neurotoxicity. Now, Stefan Schultz in Leipzig, Germany, has developed this touch evoke response assay, which is can be added to the fish embryo toxicity test. So it's basically at the end of the exposure after 96 hours, when the fish embryos have hatched out of their embryo, out of the egg, that you basically give them a little nudge and see whether or not they move or not. And then you can get some touch evoke response, EC50, that you can then include in your line of evidence. And we are still in the case at the moment where we're trying to think about whether or not we will include this in the line with the fish embryo, or to have its own separate line based on how well this assay actually performs. So we have this tool, and I would just like to try and see if I can share this with you. Hopefully you can still see my screen. Christy, could you say if you can see the website or not?
I can see your PowerPoint still.
Okay. Maybe I have to go over here then.
There we go. It's very small. Here.
Don't worry, it takes a while to come up. Okay, so this is the,
Yep.
Oops, I lost it again. This is the model that we've developed, and if you want to go in here and play with it, you can do. It has information about the project, and then under resources we have the initial FET model, and then once the new model has been updated, then we'll be able to add more to this. So it is a work in progress, but we were keen for people to try it and see how they think it works. So you have this tab here, which gives you some information about the model. And then this tab here, you can then add some information. So you obviously put your substance name in, you put in your hydrophobicity data, the molecular weight, you predict you run your toxicity on the QSAR, and then you can see whether or not you want to put if you know what the chemical category of the substance is, you can put it in here. And bear in mind that all the background here does have the original PNG database information in it. Then you can select the number of values, because you may have more than one algal toxicity data point. And then you put your EC50 values in, same for the Daphnia. And then you put your fish embryo data in. And there is a case study in here that you can load. And this is for carbamazepine. So it gives us all the information that's already in the database. So it's already preloaded. And then you just press compute. Same for if you add your own data in manually. And then you go to the results page, and this will give you your results for the different lines of evidence that's being predicted. And most importantly here, this will tell you at the top that the toxicity level of carbamazepine to AFT is most likely to be low, with a 52.2% probability. And the second most important point is that it actually does give you a conclusion on which is the most sensitive endpoint according to the data that you've input. Okay, so if I come back out of that, are we still in the PowerPoint, Christy?
Yes.
Excellent. I'm nearly up, so I'll leave this here. These links should be live. But the most important thing I wanted to say in my conclusions is that the Bayesian network and a weight of evidence approach for fish embryo toxicity data may be sufficient to be able to predict acute fish toxicity. And then the second one, I'd sort of have to bite my tongue slightly in a way, because although we've had some frustrating times trying to get this fish embryo test accepted at the regulatory level, what we've actually realized is that the FET Bayesian network actually goes beyond just being able to predict acute fish toxicity, and it improves the hazard assessment for chemicals, full stop. So I think it's a great thing that's actually come out of it, even though I've gone grey in the time I've been working with fish embryos. And then finally, I just would like to acknowledge the rest of the team. So there's some people who work at NEVA, Janneke and Raoul. We have Thomas Brownbeck from University of Heidelberg, Christian Schermer from ERWAG, Michelle Embry from Hesse, Scott Bellinger and Christine Connors from Procter & Gamble, Anders Madsen, who is the whiz with the Bayesian network, so he's from Huggin Expert, and Stefan Schultz from Leipzig, like you have said. And with that, I'll hand you back to Christy. Thank you very much for listening.
Great. Well, thank you, Adam. I have been looking forward to this presentation for a while, and you did not disappoint. Really glad to see more work being done on this endpoint. We have a few questions coming in. Everyone just input your question into the Q&A module and we can ask it. First question is there any similarity between your model and the OECD QSAR toolbox?
Well, one of the things we have considered doing is incorporating the OECD toolbox into the model, but it's very much different in a way to that, to be honest. But there are many tools that we would like to include, particularly within the first line of evidence that we would actually then be able to build on that part.
Mm-hmm.
But at the moment, we've only used the US EPA EpiSuite.
Okay, which if I think is in the QSAR toolbox.
The ECHA, yeah.
Yeah. Okay.
Which should be actually in the OECD toolbox, yes.
Yeah. Okay, next question. What about ionizable compounds? Could, under PhysChem data, could log D or pKa be added?
Yes. And that's another thing we've thought about doing. You know, at the moment, I said that first line of evidence is really just on hydrophobicity and molecular weight at the moment. But when we first started thinking about it, we were brainstorming a lot of different endpoints that we could include in there. But at the moment, for our preliminary Bayesian network, we wanted to keep it as simple as possible. But now we know that we need to include that further, yes.
Next question, is there any applicability domain that is built or is being considered? And so slightly similar question, but well, slightly different. Are these models applicable across chemical industries?
Well, yes, but we haven't proved that yet. Obviously, a lot of the work we've done is with the Procter & Gamble database, so those are industrial chemicals. But there is a drive for us to go towards some of the other types of chemicals, so pharmaceuticals, for example, because then we know they're a little bit more biologically active molecules, so that might make more of an interest. And we do have several regulatory authorities who are interested in either helping with that part where they have data, and we also have some pharmaceutical companies or pesticide companies which are also interested in supplying their data. I think the most difficult part with this is actually being able to get good quality data. And the more data we have, and as long as it's good quality, then the better it will be for us. So if there is anyone who is aware of sitting on a pile of data which maybe we could use and incorporate this, because one of the aspects to the CEFIC project is to develop this I say it very, very loosely, gold standard acute fish toxicity database, for the main reason that if we haven't got enough data to be able to compare it against, we're not going to be able to ensure that we do have a broad coverage of different chemical domains.
Yeah. Actually, that's sort of one of the questions that popped into my head was in the web demo, there's a way to select the chemical category. Does that have what is the impact of selecting that chemical category? Is that related to domain or is it related to something?
It's really at the moment only related to the fact that where we've got AFT data for those different chemical categories, and again that is based on the P&G different chemical categories that we've defined, so that doesn't really cover everything at the moment. So we need to increase that to have a broader list of different chemical categories.
Right.
And at the moment also, like I said, the different lines at the moment are all equally weighted, but one of the things we would like to do is to have them on variable weighting depending on the input and output from each of the different lines of evidence.
Okay, great. Next question, how much data curation was carried out prior to including the chemicals in the database?
Well, I won't take any credit for that. That was a lot of work that was done by Kristen Connors and the P&G team, and they've done a lot of data curation on that, yes. as I said, we have this AFT database that we are now developing or developed, and that has had a lot of data curation going into that.
Great. I think, is that there's a call right now out for data through the OECD. Is that related to building this database?
No, not as far as I'm aware. Was that through ECHA?
Yeah,
Or...
maybe I'm misremembering the details.
Well, it's always the biggest limitation. So, like I said,
Yeah.
if anyone has anything they would
We'll
like to
put it out
supply,
right
that'd be
now.
great.
If there's anyone has any data, well, it would be great if you would share it. Okay, next question. Does your model consider compounds with a short half-life that may require flow-through systems?
Short answer, no. At the moment, you know, the fish embryo test itself was originally a 48-hour test. It was a relatively static test system, but owing to the problem with the large compounds potentially not passing through the chorion, the test was extended to 96 hours, so that the fish embryos were hatched, and this is particularly important for cationic polymers, for example. But also at the same time, it was a requirement that the test had to have semi-static renewal. So the solutions need to be renewed every 24 hours to be able to give some way of being able to maintain a relatively consistent concentration. But I'd hate to have to do a flow-free fish embryo test in the whale plate, to be honest.
Okay, so a couple more questions about Bayesian networks. Do you think that this approach could be applied to predict chronic toxicity?
Yes, definitely. And that would be our next step. There are two steps we'd like to take this forward afterwards. One would be about predicting bioaccumulation, because it's the same sort of thing, the drivers that you can use for the different lines of evidence for predicting bioaccumulation would work exactly well with this. And then obviously the next step would be to include a chronic endpoint. But maybe my children or their children after me might be involved with that. I think we have a long way to go if we can't even get the fish embryo test accepted.
Well, I think this will go a long way towards it for sure. Okay, this is another specific question about the Bayesian network. Are all conditional probabilities derived from qualitative expert reasoning, or is there algorithmic inference employed?
At the moment, they are unfortunately based a lot on me and one of my research scientists who's an expert in Bayesian networks, where we had to sit down and go through the conditional probability tables manually based on expert judgment. And that was painful sometimes when you're trying to really get your head around a 5x5 table. It's quite hard work. But our next point would be that we can we've written some scripts now so that we can change the equations for each of the different CPTs, so that should make it easier in the future for us to change those things.
Okay. And also, just generally, was there a certain software that you used to construct the network?
Yes, so the Hugging software is what we use. So the Hugging expert acknowledgement I said at the end, and actually the website itself is based on the Hugging software.
Yep, I think I saw that. Okay.
We did start off with things like Netica, but then we decided to go through Hugging.
Okay. What are the cutoffs for low, medium, and high?
In terms of the okay, so what in the distribution? Well, at the moment we've put it relatively arbitrary, so we've got 100 milligrams, greater than 100 milligrams as being very high, 100 to 5 milligrams, I think it is, per liter is high, and then going down through there. So it's almost in a slightly logarithmic scale at the moment. But what we would like to do is to change the whole approach to the cutoff values to be based on individuals' requirements. So if there's a classification and labeling purposes, then maybe we just need to worry about 1 milligram per liter as being a cutoff value, for example. So that is something we were going to change in terms of user requirements.
I think it might depend on the regulatory agency.
Exactly, because this is not just going to be one model fits all. It's going to have to be one, a module which is for X, Y, or Z, depending on the different regulatory requirements.
Okay. So last question for now, unless anyone else can squeeze one in. If ionizable chemicals are to be tested in the FET, should a pH be chosen that makes the chemical as neutral as possible, or rather ionized, or dependent on cationic or anionic? If you want me to repeat that.
I don't think in the current guideline whether or not it specifies anything related to that. So I'm not sure if that's been actually considered entirely. But I will bring this up actually. That's a good point.
Is it something that's generally a consideration with aquatic testing?
I guess so, yes.
Because I wonder if it's dealt with in the guidance on difficult to test chemicals or something like that.
There probably is something in there actually about it, but off the top of my head, I would not be able to answer that.
No worries. Okay. Well, questioner suggests you look into OECD's guidance on difficult to test substances in aquatic, and we'll perhaps be able to engage further.
Yes.
Well, I actually had one more question. We have a couple of minutes left. In your model, you have a neurotox specific element. Is there a reason for that? Are neurotoxic chemicals particularly difficult to align, or?
Yeah, well, it's more based on that they can sometimes cause a bit of an outlier. You know, the difficult part, which I didn't really touch on much, was about the metabolism aspect actually, because that's where we're finding it difficult to work out how we may be able to include metabolism into the network based on that those substances which may not necessarily be metabolized in the egg, we may get false negatives from those substances. But that's causing, that's a bit more difficult for us to be able to work out what we're going to do at the moment. The neurotoxicity is just that we don't actually have neurotoxicity as an endpoint towards the FET test at the moment. And actually, ironically, I think most often the neurotoxic substances would give us a better reaction with the DAFNIT data anyway.
Okay. Great. Well, we don't have any more questions. So thank you all for listening. Thank you all for your questions leading to this discussion. You have a couple of thanks, great webinar comments coming in. So I echo that appreciation. Adam, thank you very much for your presentation.
That's no problem. And if anyone would like to get in contact to continue any further dialogue, please do so. I guess my email address will be available through this webinar.
We don't typically send out email addresses, but they'll have my email address. So
my name's not difficult to find, so
if they want to get in touch, they can always ask me.
Yes.
Okay. Well, thank you again, and for joining us on the last day of November. And I hope you all have a wonderful day and holiday. Take care.
Yes. All right. Thank you.
Bye. Thank you.